Welcome to the Diagnostic Lab for today's Explainer. I'm thrilled to have you here. As medical researchers, we aren't just looking at a patient's outward symptoms. We're diving deep inside, right down to the literal building blocks of life. By truly understanding cellular architecture, we can unlock the mysteries of human disease and pave the way for modern, life-saving therapeutics.
Okay, let's quickly run through our lab syllabus for today. We've got 6 exact diagnostic steps to cover: focusing the lens, differentiating domains, inspecting machinery, tracing origins, monitoring checkpoints, and finally, tissue pathologies. Let's get right into it.
Step 1. Focusing the lens.
Think about how wild this historical shift was. We moved from mystical vitalism to a strictly empirical, mechanistic study of cells. And none of this medical progress happens without advancements in lens technology. Back in 1665, Robert Hooke was looking at dead cork tissue under a tiny 30x microscope. Less than a decade later, Leeuwenhoek is spotting living, moving bacteria. Fast forward to 1931, Rischke's electron microscope finally lets us see deep inside to previously unresolvable internal organelles. And today, we're literally sequencing individual cell genomes to diagnose diseases at their absolute core.
And that brings us to this defining concept: omnis cellula a cellula. It literally translates to all cells arise from cells, which was popularized by Rudolf Virchow back in 1855. This was absolutely crucial because it debunked the idea of spontaneous generation once and for all. It proved life is a continuous, unbroken chain of replicating, self-contained units. For us in the clinic, it means that to figure out what's making a patient sick, we have to trace the dysfunction back to an issue inherited or acquired by a pre-existing cell.
So in our modern diagnostic labs, your workflow as students is going to look exactly like this step-by-step tutorial. First, you observe the clinical symptoms. Second, you isolate the specialized tissue samples. Third, you examine the cellular architecture under the microscope. And finally, you pinpoint the exact organelle or molecular defect that's causing the problem.
Step 2. Differentiating cell domains.
Now, before we can diagnose anything, we have to know what kind of cellular architecture we're dealing with. It basically comes down to a contrast between prokaryotes and eukaryotes. Think of a prokaryote, like a bacteria causing an infection, as a tiny, high-surface area efficiency apartment. Everything is in one open space, relying on rapid diffusion. But eukaryotic cells, the ones making up your patient's body, they're up to 100 times larger. They are multi-million-dollar mansions. And because they're so huge, they require specialized, enclosed rooms, or organelles, to keep incompatible biochemical reactions safely separated.
Step 3. Inspecting cellular machinery. If we step inside this eukaryotic mansion, we quickly see why understanding specific organelle function is basically the key to treating human disease. Take the nucleus, for example, the master control room. The cell has to safely sequester its DNA away from the reactive oxygen species that are constantly produced out in the cytoplasm during metabolism. By wrapping the DNA blueprint in a protective envelope and organizing it into chromatin, the cell ensures safe, highly organized cell division. If this room falls into disrepair, the whole mansion goes down with it.
To put this into perspective for a patient, let's look at a quick case study. Think about the lysosome, which acts as the cell's acidic garbage disposal. If a patient is missing a specific digestive enzyme in there, toxic waste builds up. That single microscopic organelle failure cascades into observable, severe clinical symptoms, leading to what we diagnose as lysosomal storage disorders, often severely impacting the nervous system. Or if the mitochondria fail to produce ATP, we see devastating degenerative diseases in energy-hungry tissues like the muscles and brain.
Step 4. Tracing endosymbiotic origins.
To really grasp how these complex eukaryotic pathologies develop, we actually have to look backward at our evolutionary history. The symbiogenesis theory, a groundbreaking concept by Lynn Margulis, is a total game changer. It explains that our incredibly complex metabolic machinery relies entirely on the ancient engulfment of free-living bacteria by a larger ancestral cell.
It sounds wild, but the evidence is literally right there inside ourselves. Our mitochondria are surrounded by double membranes, they contain their own independent circular DNA, they replicate by binary fission, and they even possess 70S bacterial-style ribosomes. Because they retain these independent characteristics, mitochondrial dysfunction acts completely differently from nuclear defects. They link directly to specific inherited diseases, usually passed down only from the mother, and it's also why certain antibiotics targeting bacterial ribosomes can occasionally cause unintended mitochondrial toxicity in our patients.
Step 5. Monitoring cell checkpoints.
So, as a medical researcher, I constantly find myself asking, what happens when our intense molecular surveillance completely fails? The visual difference you see under a microscope between healthy dividing tissue and a cancerous tumor comes down entirely to the breakdown of these regulatory checkpoints.
Healthy tissues don't just divide randomly. They follow a highly regulated biochemical clock. You've got the G1 phase for growth, S phase for DNA replication, and G2 to prep for mitosis. If DNA gets damaged, tumor suppressor proteins like p53 act as emergency molecular brakes, pausing the cycle for repairs. But in a cancerous cell, this surveillance is broken. When p53 fails or cyclins go rogue, the cell blows right past these vital stop signs, and that directly results in the uncontrolled, chaotic proliferation we diagnose as a tumor.
Step 6. Clinical tissue pathologies. Finally, we have to recognize how these specialized cells cluster together to form the actual tissues and organs of the human body. A human relies on over 200 distinct cell types, and they achieve their specialized functions by drastically altering their internal architecture. Look at the erythrocyte, or red blood cell. It literally ejects its own nucleus to maximize space for oxygen transport. A goblet cell in the gut is absolutely packed with an extensive Golgi apparatus because its entire job is packaging and secreting mucus. And a neuron stretches its membrane into massive elongated axons just to transmit signals. Form always dictates function. When we pull it all together, the cell remains the absolute foundational unit of all medical diagnostics and therapeutics. By bridging the gap between abstract microscopic structures and tangible health outcomes, we can trace pretty much every clinical symptom right back to a specific cellular or organelle failure. Whether it's a bacterial infection, a mutated p53 gene causing a tumor, or a faulty lysosome, the answers are always found by looking through the lens at the cell. I want to leave you with this final provocative thought. As we transition from traditional glass lenses to cutting-edge genetic engineering, what previously incurable diseases are we going to be able to conquer next simply by repairing the very blueprints of these cellular mansions? The future of medicine is hidden right there, inside the cell. Keep questioning, keep observing, and never underestimate the power of the microscopic world. Thanks for joining me in the lab today.